Okay, cell structure. You've probably done these models in, I don't know, junior high. I remember doing one in 7th grade. I'm going to pass out this real quick. Here. Okay, this is just, you can fill it in as we go along, or you can fill it in later, when you're watching the recorded lecture. However you want to do it. It just has all the organelles, and you can kind of fill in their function. On the back side, though, what I want you to do is just draw a line down the middle. Okay, and you're going to draw a prokaryote on one side. Draw. And a eukaryote on the other. It's going to be an animal cell. Okay, hopefully your pen works better than mine. Okay, and then for 10 points extra credit, if on Tuesday you bring in a model of a cell, and however you want to do it, you can make it out of food, or whatever you have laying around the house, as long as it's anatomically correct, bring it in, I'll give you a grade. If it's made out of food, then you can eat it afterwards or whatever. 10 points extra credit. So that'll make up a lab if you want. That'll add 10 points to your exam, whatever you want. You can glue M&Ms. What's that? You're right. I don't want to make it 20 points. Just 10. Okay? If you have 100% on everything, you can still turn it in and you get 10 extra points. Okay? Some bonus points for later. No, you can't donate. But that's very generous of you. I want to donate your points. All right. Okay, cells. Who was the first observer of cells? This is review. Robert Hooke. You guys ready? All right, and who came up with the cell theory? Schleiden Schwann. Yeah. Okay. Good. Okay, and we talked about some basics of the cell theory. There's actually 3 parts to it, and we only mentioned 2. What are the 2 parts we mentioned?
All things are made of cells.
All things are made of cells. And all cells come from pre-existing cells. Good. All organisms are made of cells. That's how we define what an organism is it has cells. And cells go through processes of metabolism. They inherit DNA from other cells. The smallest living things are cells, single-celled organisms. Bacteria are these single-celled organisms. And then the last one, cells come from the division of other cells. Okay, so those are your 3 parts of cell theory. This is a theory developed by Schleiden and Schwann. And we are kind of, I think we did 1 and 3 before. We're adding that second one in there as well. That is cell theory. Okay, cells are restricted. So one of the questions you might ask is, why are we made up of a bunch of little things? Why aren't we just made of one big cell? Has anyone given that any thought?
What? Yeah. You only have one.
Okay.
It gets messed up your done.
What if, okay, fair enough. You only have one cell. But, I mean, bacteria, there's only one of them. They do okay, right? But one of the reasons why you don't just have one big cell is because they're limited by certain functions. One of them is the rate of diffusion. Okay, and the rate of diffusion is affected by surface area to volume ratio, temperature, concentration gradient, and distance. So in this cell, let's say this is a molecule of oxygen, and this is an aerobic cell, so it needs that oxygen. And let's say that oxygen needs to go to what organelle needs oxygen. Well, we haven't gone over this yet, but what organelle needs oxygen? Mitochondria, yes. Okay. So let's pretend this C is a mitochondria. This is mitochondria in here, and this C is mitochondria here. Which cell is going to have an easier time getting oxygen to its mitochondria?
The small one.
All right, small one. Okay? Because its surface area is greater in proportion to the stuff inside it. Surface area to volume ratio. So if our cells were really, really huge, then we'd have a much harder time getting things where they need to go. Okay, and that's because of the effects of temperature and diffusion rates within the cell. Okay, that animation took a lot of time. Okay, so this is just illustrating the same point. You have two cells here. One is 1 unit and one is 10 units. And you look at their surface area. Rather than that going up 10, it goes up 100 times. And then you look at the volume. Rather than that going up 10 units, it goes up 1,000. So to the third. So as a cell gets bigger, its volume increases at a much greater rate than its surface area. That make sense? So that's why there's a size limit on how cells can function. So in order to study cells, because we cannot see them with the naked eye, we need microscopes. And who was the guy who saw the first cell? Robert Hooke. What did he use? He used a little microscope. It was similar to what we have. It was a compound microscope that had 2 lenses. But we have some more sophisticated microscopes as well. And we've already gone over this in the lab, so we won't spend too much time on it. This is our compound light microscope. What does compound refer to? Yeah, it has 2 lenses. Okay, and what does the light refer to? The light, yeah. You have a little light bulb at the bottom. It can see as small as 200 nanometers. That's the resolution. What does resolution mean? What's that? Body? Quality, yeah. So when you're talking about the resolution of your camera, it's how small the dots are, right? Basically the same thing. It's how well a microscope or an instrument can differentiate between 2 dots. Okay? It can at 200 nanometers for a compound light microscope. For an electron microscope, which shoots electrons at things and then has a little film behind it, the resolution is a lot greater. It's 2 nanometers. And there are 2 types that we talked about in the lab. The scanning electron microscope, which looks at stuff on the surface. So here's an ant. You can see all the deep ridges and different parts of its exoskeleton, see little beads in its eye. All of those things are less than or are greater than 2 nanometers. And if it's less than 2 nanometers, you aren't going to be able to differentiate between them. Okay? The transmission electron microscope is able to see things internally, so it's not doing a surface, it's actually looking at stuff on the inside. And that's what we have here. You can see the microtubules of a centriole. And that's because the electrons there's a film behind it, and the electrons go through it, and then whatever is blocked by the substance shows up dark, and whatever reflects shows up light. All right, so we break our cells into 2 different types, prokaryotes and eukaryotes. You can divide them further than that, but there are 2 main groups of prokaryotes and eukaryotes. This suffix karyote refers to the nucleus. Okay, U, that prefix means true. So these ones have a true nucleus. Pro means before. These don't really have a nucleus. They don't have a formed nucleus. So that's what the actual name is referring to. But whether you're a prokaryote or a eukaryote, all cells have DNA. They have a place where the DNA resides, and that's called the nucleoid in prokaryotes and a nucleus in eukaryotes. They have ribosomes, okay, which help synthesize proteins. They have cytoplasm, which is a fluid with solutes dissolved in there, required for homeostasis and growth and reproduction in the cell. And they have a plasma membrane, so they have something containing the cytoplasm and containing all the stuff on the inside. Okay, within the plasma membrane, it's made of a lipid bilayer. Okay, we mentioned that before when we talked about lipids. And embedded within there, you have proteins and other structures that help transport substances in and out and help identify the type of cell when you have cell-to-cell interaction. Okay? All right, so now we're going to talk just about prokaryotes. So that last slide was all cells. Prokaryotes are all single-celled organisms. So just the one cell is completely autonomous from the other cell. They do not have a nucleus. Instead, they have a nucleoid. And they have a cell wall. Okay, their cell wall is made of a carbohydrate protein complex called peptidoglycan, and that protects the cell from drying out when it's too dry, or from absorbing too much fluid when it's too wet, or abrasion by other cells. It just keeps it contained. Okay, the cell wall in plants and fungi, they also do have a cell wall, but it's made of a different substance, so it's different. We'll go over those in a second. Big thing is it does not have organelles. Okay, so it doesn't have membranes within its membrane. Everything in the cytoplasm is just free-floating, including the ribosomes. Some of them do contain some accessory structures like flagella or cilia. You see the cilia on this cell here. And there are two of our three domains are prokaryotes. The archaea, so there should be an R in there, sorry. A-R-C-H-A-E-A. And bacteria, or eubacteria. Okay, it's thought that the first living organisms were archaea. Okay, these are also the most abundant organisms. Okay, they are everywhere. Cover the earth. They cover your body. I saw someone posted a scanning electron microscope image of a toothbrush. Did you see that on Facebook about a week ago? And it showed bacteria on the bristle. It's plaque. Of course, where do you think the plaque goes when you brush your teeth? It doesn't all go down the same. Oh, hopefully most of it does. But some of it stays on your toothbrush. I liked one of the comments that said, "Ew, I'm never brushing my teeth again." Okay? Eukaryotes, on the other hand, they have a membrane-bound nucleus. So a membrane made of phospholipids, and it holds the DNA. It also has membrane-bound organelles. Not all of them are membrane-bound, but it does have them. It also has a cytoskeleton. So it has cytoskeleton are proteins within the cytoplasm. that give the cell more structure, just like your skeleton does. It guides things through the, if something is moving from one part of the cell to the other, they hitch a ride on the cytoskeleton, kind of like railroad tracks. Very important to the function of eukaryotes. They also have an endomembrane system. This is a series of membranes that can surround certain substances and put them to other parts of the cell or expel them out of the cell. And it helps to compartmentalize or specialize different areas of the cell. So just kind of like how your body is made up of organs that do different functions, that specialize on different areas, the organelles of the endomembrane system do the same thing. Okay, so here's a eukaryote animal cell, and there's a plant cell. Okay, so the main organelle, I don't know about main. The one that holds the DNA is the nucleus. Okay, in eukaryotes, the DNA is linear, meaning all the DNA is packed together, and so it makes these linear structures. And they're called chromosomes. Chromosomes are made of, or chromosomes plus the DNA. All right, this should say chromosomes equals protein plus DNA. Okay, so a chromosome has protein in there as well. And the DNA is spun around the protein.
The DNA.
Oh, no. This is something I'm talking about. Okay, chromosomes plus the protein is called chromatin. Okay? There's protein there too. This is right. Yeah, sorry. I was reading it wrong. When you have the protein with the chromosomes all packed up together, that's called chromatin, which is what it says. Okay? When you look at a nucleus, you'll see that there is a region that is darker than the rest, that little center part there, or here. That's called the nucleolus, and that's where ribosomes are being made. Ribosomes are constantly being made because they're very important for making proteins. And so there's a lot of activity there. There's a lot of enzymes and other things going on. The DNA is being unwound. And so it's darker because there's more stuff in there. The nuclear envelope has 2 lipid bilayers. Okay, so that's like 4 layers. And it has pores which control what goes in and out, what substances go in and out. Okay? You can see them here. Those little dots there are the nuclear pores. Here's a drawing of an individual pore. Okay, it controls what's going in and what's going out. Okay, ribosomes are not an organelle, but they do reside on organelles. They're made of ribosomal RNA. Go figure. They synthesize proteins, all the stuff we've talked about. And you have 2 types. Okay? These little red dots out here are ribosomes. These are called free ribosomes. And then you also have this, I don't know, what does that look like? Curvy area. This is the endoplasmic reticulum, which is made of another membrane, but it also has ribosomes embedded within it. So it's called the rough endoplasmic reticulum. It's rough because those ribosomes are on it. Okay, so you can have free or embedded ribosomes in eukaryotes. In prokaryotes, they only have free ribosomes. They don't have an endoplasmic reticulum. Okay, there are 2 types of endoplasmic reticulum. I mentioned the rough ER already, and the ribosomes give it its rough appearance. The smooth ER does not have ribosomes, okay, and it functions to compartmentalize and sort proteins, put them in little packages and send them off. Okay? It can also store calcium in your muscle cells, which is very important for the function of your muscles when you contract. Also does lipid and carbohydrate synthesis and will detoxify some substances. Okay? And it's usually connected in some way or very close to your rough endoplasmic reticulum. Everyone get that? Okay. Your Golgi apparatus is similar to the smooth ER, similar in shape, but usually in a different part of the cell. A lot of things will go from the smooth ER into the Golgi apparatus. It has two sides, a cis and a trans. The substances will come in the cis side. They will fuse the membranes here, and then they will become altered in some way or broken down in some way. And then they will form vesicles on the other side. This is a secretory vesicle. It is now ready to be transported to a different part of the cell or to be released from the cell entirely. Okay, so it's packaging things like the post office, packaging things and sending them where they need to go. That's the Golgi apparatus. Okay, lysosomes are little vesicles made by the Golgi apparatus. Okay, and the prefix lys means to cut. Okay, so they are filled with digestive enzymes, and they cut stuff up and neutralize it so it's no longer harmful. Or if it's a waste product, they'll cut it up, and then they'll recycle the amino acids or the lipids or whatever it was made of. Formed in the Golgi apparatus, what's doing the work in there are these digestive enzymes. Okay, and they're basically just these little circles found throughout the cell. When something gets old or damaged, like this mitochondria here, the lysosome will envelop it, and then break it down, recycle it. So these are the garbage guys, trash guys. They come and pick up the trash, sort it out. Perioxysomes are just like the other vesicles, only they contain specific enzymes that break down hydrogen peroxide. Hydrogen peroxide is very reactive, and it will destroy the cell if it's not taken care of. So it converts it with an enzyme called, good question. Okay. Catalase. Okay. It'll break it down and convert it into water. And it also contains enzymes that will oxidate fatty acids, okay, which is important for breaking down fatty acids for energy. Okay, vacuoles. These are found pretty much only in plants. There are some little ones in animals, but for the most part they're just in plants. And they are membrane-bound structures, usually just filled with a bunch of water. The central vacuole in plants, this whole thing here. Okay, and you can control how much water is in there, and that'll affect the shape of the cell. Also has storage vacuoles. Okay, they'll store molecules for when they're needed for energy. Some fungi and protists, again, these are not plant cells, those would be animal cells. Well, protists would be animal, fungi would be cell group. They have contractile vacuoles, so they will contract, get smaller, and help move the cell or change the shape of the cell. Okay, but for the most part, these are only found in plants. Okay, mitochondria, this is what I asked about earlier. We hadn't gone over it. But this is the site of oxidative phosphorylation or aerobic respiration, which is where we make energy, and it requires oxygen. Okay, the oxygen isn't the molecule that has the energy, but it is required to complete the process. It also has a double membrane. Its inner membrane is folded into these finger-like projections called cristae, or one would be a crista. And they have their own DNA, which is inherited from, in mammals, it's inherited from your mother. So all of your DNA is basically a clone. All of your DNA from your mitochondria is a clone of your mother's DNA. You don't get any of it from your father. Okay, chloroplasts are similar to mitochondria in their structure, in that they also have two membranes, and they have their own DNA. They have a pigment called chlorophyll, which is excited by sunlight, and it helps to capture energy from sunlight and convert carbon dioxide into organic compounds. It's found in plants and in cyanobacteria. It's important for photosynthesis. So photosynthetic organisms have chloroplasts. The inner membrane is composed of sacs called thylakoids. What was the inner membrane of mitochondria called? Yeah, cristae or crista. Yeah, so this is this homologous structure. It's the same thing. Thylakoid is a crista. Different function, but same thing structurally. All right, we mentioned this before, and this is a new slide I threw in here. Endosymbiosis is the process by which we think, or the theory, that organelles came into existence. So originally, it is thought that mitochondria, chloroplasts, and even the nucleus were separate organisms that somehow, probably one at a time, became enveloped in a larger organism or in a larger membrane. And then through their associations, they found that they were more efficient when they were working together, and that's what created then, over time, these organelles. Okay? And eventually they lost their ability to live outside the cell. So we don't find cells, mitochondria, living all by themselves. They are dependent on other functions within the cell. But originally they were probably something else, but similar. Okay, and the information which helped to develop this theory, or the evidence to support it, are that both mitochondria and chloroplasts have a double membrane, they have their own DNA, and they have their own ribosomes. So it's almost like they could be independent. They're self-contained. They have a way to gain energy. They have DNA, so they can pass their DNA onto other molecules, or when they split in half. So it seems like they were once independent, that became absorbed by other cells. And by working together, they lost their independence and are now part of our cells. You want to get that? Okay, cytoskeleton then is a network of proteins found throughout the cell. Helps it retain its shape, hold organelles in place, transports things. We have 3 different sizes. Microfilaments, which are the smallest. Micro means small, right? Microtubules, which are the largest. And intermediate filaments, which are between the two. Okay, actin is an example of a microfilament, and it will interact with myosin, which is an intermediate filament. Those are found in parallel in muscle cells, and they will interact, and that's what causes your muscles to contract. Okay, a centrosome is important in animal cells. This is the region surrounding centrioles, and centrioles contain microtubules, and this helps guide chromosomes during mitosis and meiosis. When a cell is ready to duplicate its chromosomes and then split in half, these are what's guiding the chromosomes to different parts of the cells. Plants and fungi do not have centrioles, so this is something that's unique to animal cells. Slowly. Okay, I mentioned cell walls before. We have three groups that have a cell wall: plants, okay, and protists, and they have cellulose as their cell wall. Which is what type of macromolecule? It's a sugar, carbohydrate, yes. Okay, fungi have chitin. Also, arthropods, insects, spiders, they also have chitin in their exoskeleton. Okay, these are different from the cell walls that are in bacteria, which is made of peptidoglycan. All right. Also embedded within the lipid bilayer, you have proteins, lipids, other molecules, which will extend out from the cell, out from the membrane. Okay, often made of glycoproteins. These are half or part sugar and part protein. The protein part is embedded within the lipid membrane usually. Or it can be made of collagen, which is another type of protein. Okay, and it will extend out from the cell, and this can help the cell in multiple ways. It can form just a protective barrier so nothing can actually get in and physically touch the cell. Or it can help in recognizing what type of cell it is and keeping... so it has an immune function as well. Cells that have glycoproteins that are recognized as being foreign will then be destroyed or engulfed by other cells. I altered this a little bit. So what I want you to know on this graph, I have a few things circled there, but I just circled the ones that are unique. Okay, so prokaryotes do not have an endoplasmic reticulum. All of them have ribosomes. They do not have microtubules. They don't have any of these organelles, basically. Animals are the only ones with the centriole. Chloroplasts are only found in plant cells. And plants are the only one with vacuoles. Okay, there are some eukaryotes that have vacuoles, but for the sake of this class, we're just going to say they're only found in plants. Okay, I really didn't do anything fancy, but I just kind of outlined or highlighted what was unique when you're comparing across prokaryote, animal, and plant cells. Okay, that is basically all the organelles in the cell. So you should, from now, be able to draw your prokaryote and your eukaryote. Okay, so go ahead and you can work with the buddy if you want with your piece of paper. You can look at your notes, whatever. Draw yourself a prokaryote and draw yourself a eukaryote. And if you're drawing yourself a eukaryote, you need to specify what type you're going to draw. You can do a plant or an animal. Okay? And maybe just start from memory or start by discussing with your buddy. And then start looking at your notes, kind of add information as you need it. I'll give you a few minutes to do that, and then we'll finish up the rest of the slides. Let's see if I can pause this. All right, let's draw these real quick. What do you need for prokaryote?
So what?
Yeah, cell wall, okay. What's the cell wall made out of? Stuff, yeah. It's made out of peptidoglycan. Remember that.
Oh, yeah.
Oh yeah, it's on the tip of my tongue. Okay, that'll be on the test. Okay? What's inside the cell wall?
The plasma membrane.
Yeah, so the cell wall does not replace the plasma membrane. And it may have some accessory structures, right, sticking out here, which would be what? Cilia or flagella. They usually don't have both. They just have one or the other. Okay, what's on the inside? Ribosomes? Okay, yeah, DNA in a region called the nucleoid. Okay, anything else? Cytoplasm, yeah. So that's just everywhere. Okay, these guys don't have vacuoles. Those are in plants, right? So if you're going to do a plant on the eukaryote side, so this is prokaryote. If you're going to do a plant, you could put a big vacuole in there. Let's just do a plant. All right, what else does a plant have? Okay, it has a plasma membrane. Put PM. Okay, somebody said something over here? A nucleus. What's in the nucleus? DNA. In linear structures called chromosomes. Yeah, I didn't say it, just wrote it. Okay, what about this dark part in the middle? What's that called? This marker's horrible. Nucleolus. Okay. And we kind of skipped over, there's another part on the outside, though, if we're doing a plant, right? We got the cell membrane. Cell wall. Okay? Okay, what's this cell wall made out of? Alright, cellulose. Okay, what else we got? Okay, you have chloroplasts, and if I had a green marker, I would draw them in green, but I don't. Okay? So these things, all the things, most of the things you've mentioned are what make them plants or are unique to plants. Chloroplast, the vacuole, the cell wall made of cellulose. The nucleus doesn't make it unique. What are some other things that are not unique? Okay, ER. You got two types. What are our two types? Smooth. What makes it rough? So we got ribosomes. Where else do you have ribosomes? In the mitochondria, yes. And in the chloroplast, yes. So we didn't draw mitochondria, we have mitochondria too. But also in the cytoplasm. There are free ribosomes. All right, anything else I'm missing?
What about the Golgi?
Yeah, the Golgi apparatus. Like a bunch of these flattened stacks. All right, I'm not going to draw the animal cell, but if you drew an animal cell, you'd also have your perioxysomes, your lysosomes, vesicles. Right. Okay, so this is a good exercise that you can do just with a blank piece of paper and a pencil. Start drawing your cells and see how much you can do without looking. Okay, and then use your notes, fill it in. Then start over, do it again, see what you can do without looking. Until eventually you'll be able to do it, the whole thing, without looking, and then you'll get all the answers right on the exam, right? That's true. All right, on the back, fill this out and bring it back next time for 10 points. So this is just 10 regular points, and then you can get bonus if you bring in a model.
So we just need to do the functional characteristics.
Mm-hmm. And they're all in the notes, so it shouldn't be too hard. All right, we're going to finish up. Finish up the slides. Okay, we mentioned glycoproteins, which are made of a sugary component and a protein component. We also have glycolipids, which are a sugar component and a fatty acid component. Okay, these also extend from the plasma membrane into the exterior. And then you also have these different proteins called MHC proteins. Those are the ones that are specifically used by the immune system to recognize if a cell is a foreign invader, a bacteria, something that's going to cause an infection, or if it's just a regular cell doing its job. Okay, these are how a cell will recognize, or the structures used for recognition. Okay. Okay, there are also a few structures which are important for holding cells together. Okay, so if you have two cells that are squished up next against each other, they may have these kind of Velcro extensions that hold them together, that keep things from getting in between, especially water. And these are your tight junctions. Okay? What part of your body is waterproof? Your skin. So you find a lot of tight junctions on the cells in your skin. Okay? The second one are desmosomes. These are like big rivets that are held together by proteins that extend then into the cell, but also attach to other proteins. Their function is to just hold those two cells together and just like glue them, keep them together. They're especially found in cells that have a lot of force, have a lot of impact, are jostled around a lot. So like the cells of your heart, which are expanding and contracting constantly, they have a lot of desmosomes. Okay, and the last one are called gap junctions in animal cells. Okay, and they aren't used to hold it together, to keep water out. They're actually used for signaling. So this gap junction allows sodium through the cell. And sodium is sometimes used as a signal. Okay, in a plant cell, they have similar structures that span between cells that are called plasmodesmata. Okay, this is a gap junction. But they function the same way. Okay, they're extensions of the smooth endoplasmic reticulum that then go into the other cell. All right, and we are done. Is there any questions? Okay, if you want to stay and work on your worksheet and finish that tonight, you can. If you want to take it home and do it as homework, you can do that as well. Just bring it back fully complete, and you'll get your full points. And then if you bring in a model, the model has to be good, too. It can't just be like a bunch of garbage thrown in a shoebox. Okay? You don't have to label everything, but you have to be able to tell me what you did. Like, if I point to something and say, okay, what is this? You don't say, I don't know. I just made it. Okay? Everything has to make sense, and you have to know what you did, and I'm going to quiz you a little bit on it. Okay? And we're going to show them in class. You don't have to come up and present them, but I will show them if you don't want to. Okay, have a good weekend.